Welcome everybody to the GlycoNet ACS carbohydrate chemistry joint webinar series. Today, as some of you may know, I'm Laura Mahal. I'm the Associate Director of Clinical Partnerships for the GlycoNet and also a member of ACS Carb. And it is my great pleasure today to introduce Professor Jennifer Kohler, who comes to us from the University of Texas Southwestern Medical Center. So I have known Professor Kohler for quite some time. She completed her undergraduate degree in chemistry at Bryn Mawr College and her PhD studies, which focused on the kinetics of protein DNA interactions, were conducted in the laboratory of Professor Alana Shephards when she was still at Yale. She then went to Berkeley as an American Cancer Society postdoctoral fellow, where she joined the lab of Carolyn Bertozzi. Unfortunately, I never really got to meet Professor Kohler while she was in the Bertozzi lab because I left right before she came in, but she did some really cool work there and then continued on to research in the area of carbohydrates as an independent professor. And so now her work has really opened up new ways of photo labeling with sugars, doing all sorts of really cool work in that area, and then also looking really at the role that fucose plays in intestinal health. And so I'm really happy to be listening to her talk today. Take it away, Jen.
Okay, so first of all, I want to say thank you to Laura for the kind introduction, and thank you to GlycoNet and the ACS Carb Division for putting together this great series. I think it's really wonderful that we can interact with one another in this way, especially right now, but perhaps going forward as well. It's a great opportunity. Promotes accessibility of the science to everybody. And so I'm going to tell you a little bit about some research going on in my lab that has to do with fucose. And so I'm trying to appeal to both the chemists and the glycobiologists with my slide here. So I have both the chemical structure and the glycobiology symbol there. Okay, so to start off, I'd like to talk just briefly about the intestinal epithelia. And so the cells that line the colon and the small intestine, the gut epithelial, are covered with a thick glycan coating. And I took these images from a paper published by my colleague, Laura Hooper, where they used a fixation method that preserves this mucosal layer, and then used a lectin to recognize the sugars in this layer. So you can see this dense green coating that's on the surface of mucosal cells of the gut epithelial. And this mucosal layer is super important for maintaining the interactions between the host, the mammalian host, and the microbes that reside in the gut. And not only is it important that there's a mucosal layer, but actually the structure of these glycans is quite important. And the focus of my talk today will be on a particular sugar, fucose, which is present in the gut epithelia, but not always present. And so fucosylation of the gut epithelia is induced. It's germ-free mice do not have fucosylation, but it can be induced by colonization with commensal bacteria. The level of fucosylation is regulated and it's altered by infection and inflammation. And so this fucose plays an important role in how the host interacts with both commensal bacteria and pathogens in the gut. And it's very interesting to me that the level of fucosylation varies from individual to individual, and there are genetic differences among us that will affect the amount of fucosylation in the gut. And this may underlie susceptibility to different diseases. And so what I'd like to talk about today, I'm going to have kind of three short parts to my talk. First, I want to just tell you about some photo cross-linking sugar tools that my lab developed and how we use them to discover a fucose-mediated interaction. And then I'll just, and that's published data, so I'll go through it pretty quickly. And then I'll just give you an update on where we are with that project, understanding what fucosylated glycans are recognized by a bacterial toxin that we've been studying. And then finally, I'll give you just a few slides to show you a new project that we're working on that also relates to fucose. And we're trying to understand the mechanism by which the IL-22 cytokine controls fucosylation of the gut epithelia. And so, as I'm sure all of you know, glycosylation plays many important roles, but one of which is to mediate interactions. So specific proteins, lectins, recognize specific glycan structures. But identifying these binding interactions can be challenging, both because of the typical low affinity of these binding interactions, and also because the nature of the interactions really depends on the cellular context. And if we break apart the cells to try to fish out these binding partners, the interactions are often scrambled. And so what we really wanted was a method that would allow us to capture glycan-dependent binding interactions within their cellular context. And the idea that we had was a relatively simple idea, but we thought, could we introduce a covalent bond between the glycans and the nearby biomolecules, mostly proteins, to capture these binding interactions as they are in the cellular setting. And then we could enable purification of these binding interactions and their characterization by other methods such as immunoblots and mass spectrometry. And so in order to introduce this covalent bond, we decided to prepare monosaccharides in which there was a photoactivatable crosslinker. And the one that we used is this diazarine. It's a small 3-membered ring. And when it's activated with UV light, it forms a carbene that can then react with neighboring molecules to form a covalent adduct. And so we prepared analogs that allowed us to introduce the diazarine onto sialic acid residues and on GlycoNet residues in glycoconjugates. And so today I'm just going to tell you about one project that emerged from the use of our sialic acid crosslinker. So what we found was, in order to introduce this crosslinker, the diazarine, onto sialic acid residues on the surface of the cells, the most efficient strategy was actually to prepare this analog of N-acetylminosamine, or MANAC, in which we introduce the diazarine on the N-acyl side chain and protect all the hydroxyl groups with the acetyl protecting groups. This compound readily enters cells and is metabolized in place of the normal MANAC, resulting in the appearance of diazarine modified sialic acid on the cell surface. And so in order to, we showed that we could do this, and then we wanted to see if we could use this diazarine modified sialic acid to capture glycan-dependent binding interactions. And we started with a test case that we thought we understood very well from the literature, and that was the binding of cholera toxin, which is a bacterial toxin, to its receptor on the surface of human epithelial cells, gut epithelial cells. So cholera toxin is an AB5 toxin. It has 5 B subunits that bind to the cell surface and carry in an A subunit that has a catalytic toxic activity. And we read many reviews and books that told us that the cholera toxin bound solely to this glycolipid called GM1. And so, and not only that, there was wonderful data characterizing this both structurally and the affinity of this binding interaction that cholera toxin could bind to GM1. And so we thought that would be a perfect test case for our study. And so in fact, we were able to add the cross-linking sugar to cells, allow the to be metabolized, and then add cholera toxin to the cells, UV irradiate the cells, lyse them, and perform a Western blot for the B subunit of cholera toxin. And you can see from this gel that under the cross-linking conditions, we see a mass shift of that B subunit, reflecting cross-linking, in this case to GM1. And we were able to show by mass spectrometry that this reflected a B subunit cross-link to GM1. So that was great. Our cross-linker worked exactly the way we thought it should. But we had done this experiment in Jurkat cells, which is a T cell line. And we thought, well, normally cholera toxin binds to intestinal epithelial cells. We should do the experiment there as well. But when we did that, we got a surprise. We no longer observed cholera toxin cross-linking to GM1. And instead, we observed it cross-linking to glycoproteins, which puzzled us. And so we didn't understand why we get different results in these different cell lines. At the same time, our collaborator, Ulf Everett and his group, were doing experiments in mice that lack GM1. And those mice, if they lack GM1 and GM1 is the sole receptor for cholera toxin, one might expect that they would no longer be susceptible to the toxin. And in fact, he found just the opposite. He found that the knockout mice were actually somewhat more susceptible to the toxin than wild type mice were, implying that perhaps there are molecules other than GM1 that played a role in the host cell intoxication. And so that made us really interested to find out what cholera toxin was cross-linking to on that gel. And we spent a long time kind of trying to understand what glycans cholera toxin was recognizing. And at first we thought it might be something similar to GM1, but eventually some clues pointed us to a different pathway and to look at fucose. And what we found using this metabolic inhibitor of fucosylation developed by Jim Paulson's group was that the cross-linking that we observe for cholera toxin to glycoproteins and intestinal epithelial cell lines, that cross-linking was eliminated when we inhibited fucosylation. So this suggested that cholera toxin might be recognizing fucosylated glycoconjugates. Of course, using reagents such as our photo cross-linking sugar always introduces like another variable to the reaction, and we might have unexpected effects from that. And so we also did experiments just to measure the binding of cholera toxin to these cells without the cross-linker. And again, inhibiting fucosylation caused a reduction in cholera toxin binding. We measured the ability of the toxin to enter the cells, and this was reduced when we inhibited fucosylation. And we measured the ability of the toxin to intoxicate the cells, and this was also reduced when we inhibited fucosylation. Together, these data suggested that fucose was important for cholera toxin binding to these cells, but it was surprising. And I have to say, one of the experiments that helped convince me that we were onto something that was a real effect was we did some competition assays with different monosaccharides. And you can see that L-fucose, which is the naturally occurring form of fucose in humans, could competitively interfere with cholera toxin binding, albeit at a very high concentration, this monosaccharide. But if we use the enantiomer, we no longer saw this competitive effect. So that suggested we were looking at a real molecular recognition event. And this was confirmed by studies from Udo Krengel's lab, who solved a new structure of cholera toxin. So a prior crystal structure showed that GM1 glycan binding to cholera toxin on this apical surface. But Udo Krengel's new structure showed that fucosylated glycans could bind in a separate glycan binding pocket. Okay, so taken together, these data suggested that cholera toxin could recognize fucosylated glycans, and that made us wonder if those types of molecules could competitively inhibit binding of cholera toxin to the surface of cells. And we did find that human milk oligosaccharides, when used at high concentrations, although the concentrations that they're present in milk, millimolar concentrations, can competitively interfere with toxin binding. And also these glycopolymers that contain fucose were synthesized by Nicole Sampson's lab and can competitively interfere with cholera toxin binding. So we're still trying to understand the relative roles of fucosylated glycans in GM1 in host cell intoxication by cholera toxin. And that is kind of the direction I want to talk about next. And so we wondered what kinds of glycoproteins cholera toxin was recognizing. And we took advantage of our cross-linker to isolate the glycoproteins to which cholera toxin was cross-linked. And one of the most abundant ones that we found was this one, CEACAM5, which is highly upregulated in colorectal cancer cell lines, like the cell line we were working in. And in fact, when we knocked down CEACAM5, this dramatically reduced the amount of cross-linking that we observed on the cells with these cells, but there was no effect on the overall binding of cholera toxin to the cells. And in fact, there was no effect on intoxication. So we were very confused at this point. So we knew fucose was important for binding. We found this fucosylated glycoprotein that could bind to cholera toxin, but it didn't seem to be that important for the overall binding or intoxication. And so we were a little puzzled. And we had a little bit of data to work with. One was that if we inhibited fucosylation, we could reduce cholera toxin binding. But if we inhibited sialation, the addition of sialic acid, we enhanced cholera toxin binding. And that was interesting for two reasons. One was that it argued against GM1 being the most important receptor because GM1 contains sialic acid. And it also said that using this cross-linker on sialic acid was maybe not the best tool to be using to study this problem. And so we decided to take a different approach. And this was led by Atessa Garashi, who's a graduate student in my lab. And Atessa said, I want to do a CRISPR screen. I want to get some new ideas about what cholera toxin is recognizing on the surface of cells. And in fact, Jonathan Weissman's lab had already done a CRISPR screen to look at cholera toxin binding. This was part of their CRISPR methodology development. And they had found genes involved in GM1 biosynthesis, but they had also found genes that had that were unrelated to GM1, such as this fucosyltransferase. And what we recognized is that their experiment had been done in a leukemic cell line. And Atessa said, I want to do an experiment in a colonic epithelial cell line. And she chose to do this. She chose to measure the binding of just the B subunit of cholera toxin cells, not the ability of it to intoxicate or enter cells. And she did a flow cytometry-based screen where she could look for factors that resulted in increased or decreased cholera toxin binding. And so this is some data from her initial screen. So in blue are the control COLO205 cells that she used. In red is the CRISPR library. And so she selected the cells that had lower binding and the cells that had a higher binding. And the data that she got out of the screen was just really exciting for us because many of these genes are involved in glycosylation, and many of them were genes that we hadn't been thinking about before as it being important for cholera toxin binding. So first, these are hits that result in, when they're knocked out, they result in reduced cholera toxin binding. And you can see that the ones highlighted in red have to do with fucosylation. So the number 2 hit is the GDP fucose transporter. And so we confirmed that if you knock out the GDP fucose transporter, you have reduced fucosylation and reduced cholera toxin binding to these cells. But there not only were these fucosylation genes present, the top 5 hits had something to do with glycosylation, including 2 that are related to glycolipid biosynthesis. And so UGCG is the first step in the synthesis of these glucose series glycolipids, and these can lead to the ganglioside series glycolipids, including GM1. So finding UGCG on the list could be consistent with GM1 being important for cholera toxin binding. But the other hits pointed toward a different pathway in glycolipid biosynthesis. And in our top hit actually pointed us toward the neo-lacto and lacto-series glycolipids. And in fact, when Atessa knocked out B3GNT5, which leads into the lacto and neo-lacto series glycolipids, she saw a reduction in both the production of this fucosylated epitope and in cholera toxin binding. If we look on the other side of her screen at the hits that result in increased cholera toxin binding, we again find a number of genes involved in glycosylation and genes involved in sialation. And again, remember with our inhibitor of sialation, we saw increased cholera toxin binding. And when we knock out genes involved in sialation, we see increased cholera toxin binding. And I'm sorry, I actually highlighted the wrong gene here. She knocked out the transporter, the CMP sialic acid transporter, and she sees an increase in cholera toxin binding. There's other interesting hits here. CMAS is involved in CMP sialic acid biosynthesis. SPPL3 is a protease that inactivates glycosyltransferases, including B3GNT5, the top hit on our inhibiting side of the screen. And we were really interested in this B3GALT5, which is a galactosyltransferase that differentiates between the lacto and neo-lacto series. And so this gave us the idea that perhaps these glycolipids, these fucosylated glycolipids are important for cholera toxin binding. And so I've just mapped kind of the hits here that lead us on this pathway from glucosylceramide to the neo-lacto and lacto series, and likely to the neo-lacto, the fucosylated neo-lacto series. And this part we're still trying to validate. And so while Atessa was doing those experiments in COLO205 cells, which are one colonic epithelial cell line, we had sent a separate colonic epithelial cell line, T84 cells, to Stephanie Archer-Hartman and Paris Stuasati at the CCRC. And we asked them to use mass spectrometry to analyze the glycolipids present in those cells. And interesting to us, they found basically no evidence for ganglioseries glycolipids in these cells, but found abundant fucosylated glycolipids. And although you can't distinguish the lacto and neo-lacto series from the data that the analysis they did, they are consistent with these fucosylated glycolipids that could be produced by the pathway that Atessa identified. So together, we have CRISPR screening. Our CRISPR screening validated our observation that fucosylation could affect cholera toxin binding to these cells. And the CRISPR screening, along with the mass spectrometry analysis of glycolipids, has pointed us to this neo-lacto and lacto series glycolipids as having a role in cholera toxin binding. So right now we are validating how these knockouts affect cholera toxin binding. And we want to test directly if the fucosylated glycolipids bind directly to cholera toxin, or if somehow by producing these fucosylated glycolipids, we're affecting other glycosylation pathways in a way that affects cholera toxin binding. And so that work is in progress. And the thing that we haven't really addressed at all yet is this pathway that we identified as being important for cholera toxin binding to the cell surface. Does it affect the ability of the toxin to intoxicate cells? And if so, in what direction is that effect? So that's the work that Atessa has in progress. All right. So for the last kind of little vignette, I want to tell you another story that we're working on that has to do with fucosylation in the gut. And so we got very interested in fucosylation in the gut through that cholera toxin project. And when Daniela Carroll joined my lab as a postdoctoral scientist, she wanted to really understand how that fucosylation was regulated. And there were a number of really interesting papers coming out in the years prior to that, trying to study fucosylation in the gut and the role of a cytokine, IL-22. And so I've taken this figure, again, from a commentary by my colleague, Laura Hooper, that comments on this paper from Gatto et al. In 2014. And so they worked in mice, germ-free mice, which have little fucosylation on the surface of the gut. But when they're colonized with segmented filamentous bacteria, this somehow signals to ILC3 cells to produce the IL-22 cytokine, which signals to intestinal epithelial cells through the IL-22 receptor, activating the STAT3 transcription factor and turning on transcription of genes, including FUCT2, and results in fucosylation of the surface of these cells. So we thought that was really fascinating, and we wondered if we could replicate that in an in vitro system. And so Daniela worked with two different intestinal epithelial cell lines, KECO2 BB1 cells, which are kind of similar to the small intestine, and T84 cells. She grew these cells on transwells and added recombinant human IL-22 and looked to see if there was a change in fucosylation on these cells. We expected from the prior literature to see a change in alpha-1,2 fucose, but by these lectin blots, we didn't really see much of a change. But we also, as a control, looked at alpha-1,3 fucose, and we were surprised to see that it looked like that there was an increase in alpha-1,3 fucosylation using this lectin blot. So we wanted to kind of look a little closer, and instead of using lectins, we turned to antibodies that could recognize different fucosylated epitopes. And we saw a pretty dramatic increase in production of the Lewis X antigen on glycoproteins from these cells in response to IL-22. And so this was a little surprising because the mouse data had shown that FUCT2 was upregulated, resulting in increase in alpha-1,2 fucosylation, but we were seeing this increase in alpha-1,3 fucosylation. So Daniela checked to see if this was occurring through the canonical IL-22 pathway. And indeed, if she used the IL-22 receptor, a blocking antibody against the IL-22 receptor, we saw absence of the induced fucosylation, but an isotype control had no effect. And an inhibitor of the STAT3 also blocked this induced fucosylation, so suggesting that it was through this IL-22 receptor STAT3 pathway. We also looked to see kind of what glycans the fucosylation was on. And we used this reagent from Carolyn Bertozzi's lab, STICKY, which is a mucin selective protease that will clip off mucins. And so she treated cells with IL-22, then treated them with STICKY or not, and then probes with the antibody against Lewis X. So you can see by this microscopy image that IL-22 results in an induced fucosylation of these cells, and that much of this fucosylation was removed when treated with STICKY. So that suggests that at least some of this Lewis X modification is on mucin-like glycoproteins. So then we wondered how IL-22 was regulating fucosylation, since it didn't seem to be through FUCT2 if we weren't making alpha-1,2 fucose. And we looked at a number of genes involved in fucosylation, and we didn't see any change in the transcription of these genes in response to IL-22. So we needed to get a new idea. And so we just tried an unbiased approach. We did analysis of the transcripts in the cells treated with IL-22 or not. And we got a list of genes that were upregulated in response to IL-22. And we looked to see which genes might have something to do with fucosylation. So you can see that FUCT2 is changing, but only by a little bit in these cells. But this glycosyltransferase B3GNT7 is changing quite a lot. B3GNT7 is a GlycoNet transferase that can extend polylactic chains that are found in keratin sulfate glycans. And these chains can also be fucosylated. And there was actually hidden in the supplemental of this paper, there was evidence that this also occurred. IL-22 also regulates B3GNT7 transcription in colonic enteroids, so consistent with our data. And so Daniela looked to see if transcription of B3GNT7 was regulated by IL-22 through the canonical pathway, and indeed it was. When you treat cells with IL-22, you see an increased transcript level for B3GNT7. It's blocked with the IL-22 blocking antibody and reduced when you inhibit STAT3. And this was true both in KICO2 cells and T84 cells. And she also checked to see how that changes in B3GNT7 expression affected the production of Lewis X, the fucosylated glycan on the glycoproteins in these cells. And we were only able to achieve a partial knockdown of B3GNT7 in these cells, but we did see that even this partial knockdown resulted in a reduced Lewis X expression. But perhaps more importantly, when we overexpress B3GNT7 in these cells, we see increased Lewis X expression. And this is in the absence of IL-22. So all you need to do is overexpress B3GNT7 in these cells, and you'll get more Lewis X. And so this suggested to us this model in which IL-22 treatment signals through this canonical IL-22 receptor STAT3 pathway to induce transcription of B3GNT7, which can assist in the formation of these polylactic chains, which are substrates for fucosylation. And so the ideas that we need working on now, we don't know if these are keratin sulfate type glycans that are being fucosylated. We're curious whether the expression of this gene affects the properties of the mucosal layer, and also whether the expression of this gene affects the ability of the host bacteria microbe communications, since fucose is so important for host microbe communications in the gut. So that's the directions we're headed with that. And I just like to acknowledge the people who led the projects I told you about. So the CRISPR screen that we're working on now to learn more about what color toxin recognizes on cell surfaces has been led by a great graduate student, Atessa Garashi. She got the CRISPR screening working in our lab, and she's working hard now to validate these kind of hits that are coming out. And Dr. Daniela Carroll, a postdoc in the lab, studied the IL-22 induced fucosylation. And so I'd like to thank them and the remainder of their group, and also thank our collaborators for all the work we've done together. Thank you all for your attention, and I'm happy to answer your questions.